have you ever wondered how scientists can actually grow living human or animal cells outside of the body? I mean, in a lab? Well, today we're going to take a virtual peek inside a cell culture lab to see exactly how they pull it off. Let's dive right in.
So, this is the big question, right? How in the world do we grow living tissues in a dish? It honestly sounds like something straight out of a sci-fi movie, but believe it or not, this is the real-world foundation for so many incredible medical breakthroughs. The secret? Well, it all comes down to perfectly, and I mean perfectly, replicating the conditions for life.
And you might be asking, okay, but how important is this really? Well, let me tell you, this technique is basically the bedrock of modern medicine. We're talking about everything from the polio vaccine that completely changed public health to the insulin that millions of people depend on every single day. And it doesn't stop there. Think about the most cutting-edge fields like gene therapy and tissue engineering. All of it, it all starts right here with something as seemingly simple as cells growing in a dish.
Okay, so our tour is going to start in what I like to call the command center. And in here, there is one rule that trumps all others: absolute, total sterility. You see, when you're working in the microscopic world of cells, the biggest, most terrifying danger is an enemy you can't even see. And that enemy is contamination. I'm not exaggerating when I say that just one single stray bacterium or fungal spore can completely wipe out weeks or even months of painstaking research. It's devastating. So the entire lab is basically designed as this intricate fortress to keep those invaders out. So the question is, how do they actually win this invisible war? This is how you win. It's called a laminar airflow hood. And honestly, the best way to think about it is like a sterile force field for your cells. It's constantly pushing out this invisible curtain of ultra-purified air, thanks to something called a HEPA filter. This creates an absolutely pristine workspace where nothing unwanted can get in. This single piece of equipment, it is, without a doubt, the cornerstone of the entire lab. Okay, so a sterile environment is absolutely crucial, but it's really just the beginning. Once you've kicked all the invaders out, the next big challenge is to build the perfect, most comfortable little home for the cells to live and grow in.
And it turns out, cells are, well, they're pretty particular tenants. They have three non-negotiable demands. First up, they need a constant, super precise temperature, right around 37 degrees Celsius, normal body temp. Second, they need a stable pH, just like what you'd find in our bloodstream. And the way they do this is pretty genius. They use carbon dioxide. And finally, third, they need really high humidity, about 95%, to make sure they never, ever dry out.
And the machine that makes all this happen? It's the CO2 incubator. Seriously, if the hood is a force field, then this thing is a 5-star luxury hotel for cells. It flawlessly maintains that perfect trifecta of temperature, humidity, and CO2 levels. It creates this incredibly stable, life-sustaining atmosphere that the cells need to not just, survive, but to actually thrive. Okay, so we've built them a super safe, super cozy home. But how do we actually check in on them? I mean, how can we be sure that they're healthy and happy in there?
Well, scientists do daily checkups using a really cool tool called an inverted phase contrast microscope. It's inverted because it lets them look up through the bottom of the dish without disturbing anything. They're looking at the cell's health, their shape, and they're also measuring something called confluence. That's basically just a fancy word for how much of the dish's surface the cells have covered. Once they hit that sweet spot of about 70 to 80%, it's a signal that they're getting a bit crowded and it's time to move them into a bigger home.
Now, a perfect house is great, but it's not everything. Our cells need to eat. So what's on the menu in this tiny little cellular metropolis? Let's break down the recipe for life. So every meal starts with a foundation called basal media. The best way to think of this is like the nutrient-rich broth of a really good soup. It's usually this vibrant pink or red color, and it is just packed with all the essentials. We're talking salts, sugars like glucose for energy, amino acids for building proteins, and all the critical vitamins. But here's the thing. That broth alone isn't quite enough. The cells need a signal, an instruction, that tells them to actually grow. And that's where the secret sauce comes in. It's called fetal bovine serum, or FBS for short. In the lab, this stuff is basically treated like liquid gold. Why? Because it's this incredibly complex cocktail of over 1,000 different growth factors, hormones, and proteins that essentially gives the cells the green light to start dividing and multiplying. And if you look at the final recipe here, it's pretty interesting. The vast majority of it, like 89%, is that basal media broth we talked about. The FBS, that liquid gold, only makes up about 10%, but its impact is absolutely massive. That tiny little fraction is what provides all those crucial signals that drive the entire culture forward.
All right, to complete the perfect meal, a few more key supplements get added. First, you've got L-glutamine, which is basically an extra energy boost for the cells. Then you add a cocktail of antibiotics, usually penicillin and streptomycin, which acts as a kind of insurance policy against any sneaky bacteria. And finally, something called phenol red gets mixed in, and this is super clever. It's a pH indicator that acts like a visual alarm system. If the conditions go wrong and become too acidic, the media will literally change color from red to yellow, telling the scientist something's up.
So we've got the pristine environment, we've got the perfect meal plan. Now for the final leg of our tour, let's look at the specialized tools and vessels you need to handle life with this kind of incredible precision.
What's really interesting is that not all cells grow the same way. Most cells are what we call adherent. This means they actually need to attach or adhere to a specially treated plastic surface to grow. It kind of mimics how they'd exist in our body tissues. But other cells are grown in suspension, where they just float around freely in the liquid media. This method is perfect for when you need massive scale production, like if you're manufacturing huge batches of antibodies for medicine.
And of course, the daily workflow requires a very specific toolkit. You've got phosphate buffered saline, or PBS, which is used for gentle washing. There's an enzyme called trypsin that helps detach those sticky adherent cells from their flask. For long-term storage, cells get frozen down in liquid nitrogen at a mind-boggling -196 degrees Celsius. To count them precisely, scientists use a special gridded slide called a hemocytometer. And naturally, you have serological pipettes for moving all these liquids around with pinpoint accuracy.
So we've seen the whole setup, right? The sterile hoods, the 5-star incubators, the complex recipes, the precision tools, it's a lot. So let's just pull back for a second and ask the big question. Why does mastering this microscopic world matter so much?
You know, this quote really gets to the heart of it. The whole point of this entire incredibly meticulous system is to get results that are reliable, reproducible, and scalable. In other words, you want to be able to trust your results, you want other scientists to be able to get the same results, and you want to be able to scale up your work. It's all about turning biology from something that can feel a bit unpredictable into a precise, dependable science.
And believe me, the margin for error here is razor thin. For a cell culture to even be considered a success, more than 90% of the cells have to be viable, meaning they have to be alive and healthy. What that tells you is that every single step in this incredibly complex process, from the air filtration at the beginning to the exact food recipe, is absolutely critical. You have to get it all right to meet that super high standard.
And really, this brings us to the most exciting part of all. We've developed this truly incredible ability to sustain and grow life in a dish with a level of control that is just breathtaking. So now the question isn't really, how do we do it anymore? The real question is, what are we going to do with it next? Armed with these powerful techniques, just think, what monumental medical breakthroughs are waiting for us right around the corner?